Welcome to Chapter 15, Part 3, the Special Senses. In this section, we're going to talk about the ear, hearing, and balancing. Now, our ear function is, part of their function is obviously hearing, but also balance and equilibrium. Inside of our ears, deep inside the cochlea, which we will talk about later on in this section, we have something called mechanoreceptors that specifically detect sound waves. Sound is, take a look at this picture here. When you hear something like my voice now, or music, or any sound, it is coming in as an actual wave. Even though it's invisible, it's still a mechanical force. So we have what are called mechanoreceptors. Each of these mechanoreceptors deep in the ear consists of these specialized hair cells. Okay, they have microvilli that can detect, and they're very sensitive to those wavelengths that come in. And so they pick up the wavelengths and see how these little microvilli are moved, and that's demonstrating that they're sensitive, or they are responding to the stimulus, the sound wave, which is, in this case, the stimulus is a sound wave. All right, now when sound waves strike the ear, they create a vibration, and eventually that vibration will be transmitted to the brain for processing. So let's see how that happens. We're going to have to start with the basic anatomy. So the human ear, okay, we have the outer ear that extends this way, the middle ear, which extends to here, and the inner ear. Now, I'm going to go through the full anatomy. This is very, very basic, but I just want to start with this. Okay, the outer ear. We have this kind of hook-like cartilaginous part of your ear is called the pinna. Okay, and your earlobes down here, this kind of fleshy part. Part of the outer ear is this canal called the auditory canal, or the external acoustic medius. And then we have this little tiny membrane called the tympanic membrane. Right there. This is your eardrum. The tympanic membrane separates the outer ear from the middle ear. And there's more anatomy that we're going to learn, but for now, we're going to start with this very simple anatomy. Now, inside the middle ear, there are three tiny little bones. The malleus, right here, incus, and stapes over here. Okay, this one. That's one bone. And see how they're all touching? And notice that the first one, the malleus, is connected to the eardrum, the tympanic membrane. And also, an important thing to notice is, let me just remove some of this. Okay. See how they're both connected? The malleus is connected to the tympanic membrane on this side, and the stapes is connected over here. Okay. And it's specifically connected to the oval window, which is not present in this picture. Okay, inside of the middle ear, this is air-filled, and notice that it's connecting to the auditory tube, which connects to your throat. That's why the ear, nose, and throat go together. And then we have this little round window, and then the inner ear. The inner ear, the primary organ, is the cochlea, this little guy that looks like a snail. But there's more than just the cochlea. We have the semicircular canals. See these little semicircles? And they are indeed canals. Okay, and then we have the vestibule, which is not visible here, and we have the vestibular nerve, the cochlear nerve, and these nerves go and connect to the brain. So when a sound wave comes in, I'm going to close this and then open it back up, so just hang tight with me. Okay, so when a sound wave comes in, it's going to come in this way, and then it's going to get funneled or concentrated through the auditory canal. And then that sound gets amplified because as soon as it hits the tympanic membrane, it's going to cause the membrane to vibrate back and forth. And because the tympanic membrane is connected to these bones, malleus, incus, and stapes, the vibration is going to continue. It will move from the tympanic membrane, which will vibrate the malleus, which will vibrate the incus, which will then hit the stapes, which will then hit the oval window, which will be transmitted into the cochlea. And we'll talk more about the details of that. And the cochlea is capable of interpreting that information and sending it to the brain for processing. Okay, that's a simplified version of the explanation of the process of hearing. Now let's get into the full anatomy. Now the ear has three regions, as I said: outer, middle, inner. And ultimately, the outer is air-filled, the middle is going to be air-filled, and the inner is filled with fluid. And this is important because this fluid carries those vibrations that we discussed earlier, and it's going to be able to turn and help change those mechanical vibrations and transmit them into something electrical at some point. Now, the structure of the ear, the outer ear, and the middle ear and the internal ear, they each have a job. The external ear is just for hearing only. The middle ear is also for hearing, and the internal ear is for hearing and equilibrium, because the internal ear has receptors for both hearing and balance. And they respond to different stimuli based on stimuli for hearing, stimuli for balance, and the inner ear works to keep you from having vertigo, for example, keep you balanced, keep you from getting dizzy, things like that. That's what we mean by maintaining equilibrium and balance. Okay, now the outer ear technically has two parts. And so it's the auricle / pinna, which is this fleshy part, right? And notice that the pinna is this shell, is just this part, the shell-shaped part, right? This one. Okay. The helix is the cartilaginous rim, and then the lobe is down here. The second part of the ear is the external acoustic medius, okay, which transmits sound waves to the eardrum, which is the tympanic membrane. Okay, the tympanic membrane, also known as the eardrum, it separates the external ear and the middle of the ear. It's this tiny, thin, translucent connective tissue that responds to vibrations. We're going to zoom in and check it out. Look at that. That's the tympanic membrane. So as soon as those sound waves are coming through, and they hit this thing, it will begin the transmission of sound by mechanical stimulation. Okay, so when the tympanic membrane transfers those vibrations to the three ossicle bones, the malleus, incus, and stapes in the middle, what happens next? All right, so let's review the outer ear and then we'll move on. The primary function of the outer ear is to channel sound waves to the middle ear. That's its job. Now we have the pinna, the auditory visible portion of the ear, the auditory canal, and the tympanic membrane that separates the outer from the middle ear. The middle ear, its function and job is to amplify sound, make sound louder, to take those vibrations and amplify them. Inside of this air-filled space, we have three bones. They're called the ossicles. The malleus, which is also called the hammer, incus, the anvil, and stapes, which is referred to as the stirrup because it looks like the stirrups that go on a saddle, a horse saddle. And ultimately, the sound waves cause the vibrations of the three bones in sequential order until it enters the cochlea. Now, ultimately, that will amplify the sound. The bones will amplify the sound significantly before it's transferred. Okay, I said this in the previous slide, but I'll say it again. The auditory tube connects to the throat. I may not have mentioned this, but it does equalize the pressure in your ear canals. All right, now the internal ear. The internal ear is quite unique. It's referred to as a labyrinth because it is quite a maze. It's located in your temporal bone behind your eye socket. So remember the temporal bone and where your eye socket is, okay? And remember how the temporal bone has that hole? Of course, it connects to this eventually. Okay, there's three regions of the internal ear: the vestibular region, semicircular canals, the cochlea. And the inner ear is filled with fluid, paralimb fluid, which is similar to the cerebral spinal fluid. Okay, so this snail-like structure, okay, right here, that is the cochlea. The vestibule. Okay, so kind of these little egg-shaped structures here. Okay, and then the semicircular canals are indeed semicircles. All right, so let's take a closer look. Inner ear, the cochlea. Let's start with that. That cochlea, so I want you to take a look at this picture, this cross section. See how they've cut? They've taken a cross section and cut the cochlea open so we can see the internal anatomy. Now, the primary function of the cochlea is to convert sound waves into an action potential, which is going to, an action potential is an electrical stimulus. Now, if you remember from human bio, when you guys talked about neuro, if you did take that class, if not, just know that an action potential is synonymous with firing off a neuron. So it's going to take mechanical sound and convert it into electrical. So it's going to take mechanical stimuli and convert to electrical / nerve impulse. Okay, that's quite a talent to be able to do that. So the structure of the cochlea, it has two major parts. Okay, the coiled taper tube and the organ of Corti. Now, if we take a close look at the coiled taper tube, we can see that ultimately what we have here are two canals, okay, the vestibular canal and the tympanic canal. So here's one, the vestibular one, the tympanic one right here. Now notice this is inside of every single one of these. So see, there's a cochlear duct, vestibular membrane, vestibular canal. All right, now the organ of Corti is located in the cochlear canal. So it's going to be in here, and actually specifically right there. See that little guy? We're going to zoom in on him. That's the organ of Corti. Inside of that organ, there's hair cells, mechanoreceptors, that can pick up those waves because it has a tectoral membrane that can detect mechanostimulation. So this is a closer look at the organ of Corti, and we're going to go a little closer actually. There we go. Okay, so when vibrations are coming through, they're going to pass through the from the stapes to the oval window through the cochlear fluid, and it's going to create these pressure waves. Pressure waves are going to cause these cells to these hair cells are going to feel it, detect it, pick it up, and because they are pushed and bent, it will create a nerve stimulus that will get picked up and carried by auditory and vestibular nerves to the auditory cortex of the brain, where the brain can indeed interpret exactly what it is you're hearing and convert that sound into meaningful information. You look closely at the anatomy of the cochlea under the microscope. See these little hairs? Those are the hair cells. Now, these hair cells can be damaged, and if they are damaged, let's say they're damaged, this will affect hearing drastically. Now, going through the anatomy of the inner ear is important. So you want to know all of it. You want to know that the oval window is back here behind the stapes, okay? And you want to know the cochlea, the round window, the vestibule, both of them. This one is called the sacral vestibule, and then this one, this bigger one, is the utricle vestibule. Okay, the semicircular canals, both anterior, posterior, and lateral. Okay, anterior is the first one. Posterior is towards the back. Lateral is towards the outermost region. Okay. Now look at all these nerves that are involved. It's just not just one. It's the cochlear nerve. Okay, then we have the vestibular ganglion, the inferior and superior vestibular ganglion. Then those connect to the vestibular nerve. Then we have the facial nerve. A lot of nerves involved in this process. So what we just discussed was the cochlea. What about the vestibular apparatus? Okay, let's take a look. Inside of the vestibular apparatus, that would be here and then here too, we have these two membranous sacs. Okay, the utricle and the saccule. The saccule one connects to the cochlear duct, and the utricle one connects to the semicircular canals. And ultimately, inside of these regions, we have membranes, hair cells, supporting cells, and vestibular nerves. So ultimately, on top of all of that, we also have receptors that respond to gravity and changes in position. So see this woman? She's got her head straight here, okay? And therefore, all of this should be straight as well. Okay, when she bends her head this way, notice how the flow of the membrane will change. And these, and the cells will move in tandem. And basically what they're doing is they allow the detection of the change in head position, and it processes the linear acceleration of gravitational movement, therefore balancing the body and maintaining equilibrium. So if you turn your head left, right, down, back, you don't get dizzy because of this balance. Okay, so let's take a closer look. Now, we have receptors called the macula. This is a receptor region that responds to movements, okay? And so if your head's upright, gravity's going to pull things downwards always, right? And there's a steady stream of action potentials that are coming through, no big deal. Okay, you move your head forward, tilt it forward, it's going to bend these hair cells, and they will depolarize and stimulate these nerve fibers, okay, increasing action potentials to the brain so that the balance is kept. If you tilt backwards, it's going to exert force this way, and the hair cells will bend that way, hyperpolarizing these cells and decreasing action potentials, thus maintaining your balance. Now the next thing we're going to talk about are the semicircular canals. There's 3 that we've discussed already, but what we haven't discussed is their arrangement. That each one is arranged in a dimension of space to maintain your equilibrium. So they also contain hair cells. So if we look deep into the inner ear, okay, we have these receptors called the ampulla. They are inside of the semicircular canals. One is in each, or there are actually more than one in each, but either way, that's where they're located. And so ultimately inside of there, we have the hair cells, okay, and they're surrounded by this material. It's like a gel-like matrix, but it's inside of a membrane, and the whole thing is called the cupula. Now, when you rotate your head, like this guy right here, okay, the fluid that is in the semicircular canals bends the hair cell, these hair cells, okay, in the opposite direction, and allowing this enlargement of the ducts of each ear canal. And ultimately, this ampulla housing these receptors is going to allow these receptors to respond to angular rotation of the head, sending that information to the cerebellum of the brain, and allowing your body and brain to maintain equilibrium as well. Okay, quick recap of the inner ear. Semicircular canals, their job is to maintain equilibrium and to assist in rotation, and maintain your equilibrium when you rotate your head. Okay, the vestibule is involved in equilibrium, but specifically head position relative to gravity, and if you move your head forward, linear acceleration. And the cochlea is specifically for hearing. Now, let's talk about hearing. Now, in our auditory canal and the middle ear, sound waves enter that auditory canal. They hit your eardrum. That eardrum vibrates. Okay, that vibration is amplified across the ossicles, the middle ear bones. That hits the stapes, which hits the oval window. The oval window transmits that vibration to the cochlea. And then the cochlea will send that to the brain. So if we look closely here, you can see the waves coming in at step 1. Sound wave vibrates the tympanic membrane. The vibration, the auditory ossicle is number 2. Follow the numbers as I go through. Number 2, right here. Okay. Auditory ossicles vibrate. So these are the ossicles. Malleus, incus, and stapes, the 3 ear bones. Okay, they vibrate, and the pressure then is amplified. 3, pressure waves created by the stapes. So the stapes hits over here and creates these pressure waves, pushing across the oval window. The oval window is right here. We can't see it. The stapes is connected to it, but it is there. So let me just delete this. It's behind the stapes. Either way, the oval window will move, and it'll cause the movement through the fluid. And that movement through this fluid is going to allow sound frequencies to travel through and hit those hair cells. Okay, now once that sound comes through and the sound frequencies excite those hair cells, the sound range through the cochlear duct and the vibrating membrane is going to occur. It's going to vibrate this basilar membrane in the cochlea. It's going to vibrate through the cochlear duct. And ultimately, once those hairs, the inner hairs, pick that information up, they'll send it off to the brain. Okay, now what about hearing disorders? Deafness, for example. This is what hair bundles or hair cells should look like in the ear. And if after loud sounds, they can look like this. And these are damaged. This can cause loss of hearing. It can basically, deafness is usually because of damage to those hair cells. It can happen if there's damage to the tympanic membrane or any of the bones, any of the bones in the ear, anything that impedes those sound waves can affect hearing. Okay, otitis media is when there's inflammation of the middle ear, which you can see in this picture here, normal versus inflamed. Meniere's syndrome is when the inner ear is impaired, and it's associated directly with hearing. And basically what happens is this fluid that's inside is backed up, leading to pressure, distorting the ability to hear and remain balanced. Okay, we're going to watch a video that is incredibly significant, so stay tuned for that.
this video, we're going to talk about our sense of sound. Our sense of sound is also known as audition. So in order for us to hear anything, two things have to happen. First, there has to be some sort of stimulus, and in the case of sound, that stimulus is something known as a sound wave. So a pressurized sound wave. So that's the first thing that needs to be present in order for us to hear anything. The second thing, aside from the stimulus, is some sort of receptor that's sensitive to the stimulus. And in the case of audition, that receptor is something known as a hair cell. And the hair cell is a specialized receptor that's found within the cochlea, and we'll discuss this in further detail later in the video. So let's take a look at these two things. So what exactly is a pressurized sound wave? Let's look at an example. So whenever you clap your hands, you have learned from experience that people, when they clap their hands, it makes a sound, and it makes a very distinct sound. So let's imagine that these two lines right here are your hands. And when you clap your hands, the lines move towards each other. So your hands are moving towards each other. And they're moving towards each other fairly quickly. Now, in between your hands are a whole bunch of little air molecules, which I'm drawing. And these air molecules, which I'm drawing right now, are these little, let's imagine that they're these little purple dots. So in between your hands are a whole bunch of these air molecules. And so they're just floating around, doing their thing. And then all of a sudden, the hands are moving towards each other. And so all of a sudden, the space that these air molecules occupy gets a lot smaller. So a little bit later in time, as the hands are moving towards each other, so here we are just drawing the hands almost about to touch. So what happened was all these air molecules that were just floating around, they had all this space. Now all of a sudden they're really compressed. So they're really, really close together and they're super compressed. They're very compacted now. So you can imagine that as your hands are even closer together, that the air molecules get even more compacted. And so basically what is effectively going on is the air molecules here are getting pressurized. So as you bring your hands together, you're actually adding all the molecules up, and it creates this pressure. So this area of pressure actually tries to escape. So it tries to escape, and it kind of goes this way. It tries to escape out wherever it can. And so as it's escaping, it creates these areas of high and low pressure, and that's what I'm representing here by these lines. And these areas of high and low pressure are what are known as sound waves. So we could have different types of sound waves. We could have sound waves that are really, really close together or really far away from each other. And so if we draw this graphically, it might look something like this. So basically what I'm drawing here is up here would be an area of high pressure, over here would be an area of low pressure, and so basically there are just areas of high and low pressure. And how close these peaks are together is the frequency. So if I clap my hands even faster together, or if there's something else that's a higher frequency, a higher pitch sound, the sound waves would be closer to one another, and it would look something like this. So depending on the frequency of a sound wave, it's perceived to be a different noise. So let's imagine that this sound wave right here is F1, and that this one over here is F2. Now, sound waves of lower frequency actually travel further. So this is also, this actually happens in the ear. So these lower frequency sound waves actually travel further, and they actually penetrate deeper into the cochlea, which is a structure that we'll talk about in a little bit. So if you look at these two different sound waves, they both have different frequencies, but you might have noticed, so let's imagine that this frequency F1 is generated by hands clapping and F2 is generated by somebody talking. So you can actually listen to both somebody talking and clapping their hands at the same time. And what that would look at like if we were to add these two frequencies together would be something kind of weird. It might look like this. It's not very uniform. So if you were to add the two frequencies together, you'd get this really weird jumbled frequency, which we will label F3. Now, your ear has a very difficult task now because it has to actually break this frequency F3 down into the two simpler frequencies F1 and F2. And your ear is actually able to do that. And it's able to do that because the sound waves actually travel different lengths along the cochlea. So now that we've talked about what a pressurized sound wave is, let's look at the hair cells, and in general, let's look at the anatomy of the ear. Okay, so here we have a diagram of the ear. And like I mentioned before, there are sound waves that will be coming in to the ear. So imagine that I clap my hands. Now sound waves are going to travel through the air and they're going to go towards your head. And the very first thing that they hit will be this outer visible part of the ear. So this is when you look at someone's ear, this is what you see. And this outer visible part of the ear is something known as the pinna. Something known as the pinna. So these sound waves get funneled by the pinna down into this smaller structure known as an auditory canal. And this is also known as a external auditory meatus. So I'll write that down here. So external auditory meatus. So these sound waves travel down the external auditory meatus, and the next thing that they hit is the eardrum or tympanic membrane. So the next thing that they hit is the eardrum. Now, what the eardrum does is it actually starts to vibrate. So as this pressurized sound wave hits the eardrum starts vibrating back and forth. And when it's vibrating back and forth, it actually causes these little bones. So there are 3 little bones here, 1, 2, and 3. And it causes these 3 little bones to vibrate. The first bone is known as the malleus, the second bone is known as the incus, and the third little bone over here is known as the stapes. So let's just recap real quick. So the sound waves come in, get funneled by the pinna into the external auditory meatus, otherwise known as the auditory canal, then hit the eardrum, otherwise known as the tympanic membrane, and the eardrum starts to vibrate back and forth. And this vibration causes 3 little bones known as the malleus, incus, and stapes to vibrate back and forth accordingly. So the next thing that happens is the stapes is attached to this oval window over here. So it's known as the elliptical window, which I'm underlining here. It's also known as the oval window. So this oval window starts to vibrate back and forth as well. So the next thing that happens is there's actually fluid. So this structure that the oval window is attached to is known as the cochlea. So this round structure right here is known as the cochlea. So inside the cochlea is a bunch of fluid. And as the oval window gets pushed inside and outside of the cochlea by the stapes, it actually pushes the fluid. So it causes the fluid to be pushed this way. And it causes the fluid to go all the way around the cochlea, and then it just keeps going all the way around the cochlea until it reaches the tip of the cochlea. And when it reaches the tip of the cochlea, what does it do? Well, the only thing it can do is go back. And so now the fluid's going to have to go back. So let's just follow this green line over here. So the fluid moves back towards where it came, but it actually doesn't go back to the oval window. It actually goes to this other window known as the circular or round window. Oops. Let me just fix that. So it goes to this circular or round window. And it causes the round window to get pushed out. So this basically keeps happening. So the fluid moves all the way to the tip of the cochlea, all the way back out, and back and forth until the energy of this sound wave actually causes eventually the fluid stops moving. All that energy is dissipated. And meanwhile, hair cells inside the cochlea are being pushed back and forth, and that transmits an electrical impulse via this auditory nerve to the brain. Now, the reason that the fluid doesn't move back to the oval window when it goes to the very tip of the cochlea is because in the very middle of the cochlea, is a membrane. Actually, let me use a different color. So there's actually a membrane. So I'm going to use this black line to kind of demarcate the membrane that runs along the length of the cochlea. So this membrane is something known as the organ of Corti. Let me just write that down here. Organ of Corti. Now this organ of Corti is actually composed of 2 different things. It's composed of something known as the basilar membrane and another membrane known as the tectorial membrane. Tectorial membrane. So one final thing that I just want to touch upon is a general classification of the different parts of the ear. So this outermost part of the ear, so from the pinna all the way to including the external auditory meatus, so all the way to the tympanic membrane and including the tympanic membrane or eardrum, is known as the outer or external ear. So next, from the malleus all the way to the stapes, so these three bones are known. So from the malleus, incus, and the stapes, that is known as the middle ear. So it looks like there's overlap, but I'm just trying to include this word over here. So the middle ear is actually this region from here to here is the middle ear, whereas the external ear is the region from here all the way out here. And then the third section includes the cochlea and the something known as the semicircular canals, which we didn't talk about, and that is known as the inner ear. So this is the inner ear. So those are the three different general classifications that we could break the ear, different structures of the ear down into. And so next what I want to do is look at the organ of Corti, or look at the cochlea a little bit more carefully, and look at how the fluid motion inside the cochlea actually causes hair cells to fire a neural impulse to the brain, which can then be interpreted as sound. Okay, so now let's take a closer look at the cochlea and the inner ear. Let me just draw a little cochlea. So it's this round snail-like structure, but let's go ahead and unroll it. So let's just unroll it and lie it flat. So if we unroll it, it'll look something like this. It'll look something like that. So basically this is just a flattened cochlea, and so you've got this little bone that we were talking about earlier, which is called the stapes. So you've got this little bone right here. It's called the stapes, and this stapes is connected to the other two bones and then the eardrum. So it's basically moving back and forth at the same frequency as the sound wave that's making the eardrum move back and forth. And this stapes is connected to this little oval membrane called the elliptical window. And the elliptical window gets pushed in and out as the stapes moves back and forth. So as the elliptical window gets pushed in, there's fluid inside the cochlea. So we've got fluid inside, and that fluid basically gets pushed all the way around the cochlea and then comes back around. And as I mentioned before, the reason that the fluid flows in this direction is because there's actually a structure right in the middle. There's this little structure, and we call it the organ of Corti. So this organ of Corti kind of splits the cochlea in two, and the fluid can only flow in this direction. So when the fluid gets over here, there's this little round window, it's actually called the circular window, and the circular window gets pushed out a little bit as the fluid kind of compresses it. So now you've got fluid flow back and forth around this organ of Corti. So what we want to look at now is we just want to look at a cross section, just a little cross section of this organ of Corti, so that we can kind of understand what happens. How does it turn this fluid motion into sound? So if you actually kind of look at a cross section, what you would see is something like this. So something like this. So you've got an upper membrane and a bottom and a lower membrane, and you've also got these little hair cells. So you've got little hair cells with little shark fin looking things on the tops. So you've got these little hair cells, and basically as there's fluid flow around this organ of Corti, it goes like that, and then the fluid kind of comes around and goes this way. So as we have fluid flow, it actually pushes down on this membrane and pushes up on this membrane. So you can kind of imagine how this fluid flow works. So as this membrane gets pushed up and down, it actually causes these little hair cells to move back and forth. So they're moving back and forth, they're vibrating, and basically what we can do is we can blow up these hair cells. So we can blow them up and look at them in a little bit more detail. So it kind of looks something like this. So there's their shark fin part, and then there's the cell. So this shark fin part actually is called the hair bundle. Hair bundle. And these aren't actually hairs. What they are is they're just a bunch of little filaments, and let me just draw it in a little bit bigger. So if we were to look at just the hair bundle, it would look something like this. It would look something like this. And each one of these filaments is called kinocilium. Kinocilium. So basically, whole bunch of these little filaments are attached to one another and they form the hair bundle. So each kinocilium is actually connected to one another by this little spring-like structure called a tip link. So it's a little spring-like structure, and each one is called a tip link. So it links the tips of the kinocilium. So if we were to actually look at a tip link, so let's go ahead and look at just this, the tip of this kinocilium. If we were to look at just the tip of this kinocilium, it would look something like that. And you've got the little tip link attached to it. So you've got this little spring-like structure attached to the tip of this kinocilium. And in fact, the tip link isn't attached to the kinocilium directly, but it's actually attached to the gate of a potassium channel. So there's the little gate right here. So this is the little gate of a potassium channel. And so as these hair cells, as the little kinocilii get pushed back and forth because the fluid is moving in the cochlea, as they get pushed back and forth, it actually stretches on this spring. So let's say that the kinocilium gets stretched. Sorry, we're going to use this color over here. Let's say that the kinocilium gets stretched. It actually kind of looks like this. So now it's getting stretched, and as it gets stretched, it actually opens up this gate. So as the potassium channel gate opens up, there's potassium outside that then flows into the cell. So you've basically got potassium out here flowing into the cell. And there are actually all these other little channels, calcium channels, that get activated when potassium is inside the cell. So now you also have calcium flowing into the cell. So the flowing of potassium and calcium into the cell basically causes the cell to fire an action potential. So it basically stimulates another cell, which is known as a spiral ganglion cell. And the spiral ganglion cell then activates another cell that is part of the auditory nerve, which then goes to the brain. So basically this goes to the brain. So this is what happens when a sound wave comes into the ear and then gets transmitted into a neural impulse by these little hair cells.